So thanks everyone in this presentation. Is some recent studies we have conducted assessing what's the after effects of ayahuasca intake and how these may be related to its therapeutic potential. So most of what I'm going to show has not been published yet, and also some exciting, what I think are exciting new findings of studies we have conducted in vitro in stem cells, which wow, I was left astonished when I saw the findings. So I hope you find them also interesting. So I have no conflicts of interest to declare. But I have people I would like to thank, especially Amanda Fielding and the Beckley Foundation for intellectual support and also for funding the studies that I'm going to show. And also very specially to this research team. They are part of the Spanish National Research Council, a laboratory in Madrid, who have conducted the in vitro studies that I will show at the end of my presentation. So, as you may know, we recently published a study in depressed patients that was conducted in Brazil. I was an advisor in this study. I was not directly involved with the experimental execution of this study, but we were very happy to find positive results here. That it was a small sample of depressed patients, around 6. It was published in the Brazilian Journal of Psychiatry, but it immediately got a lot of international attention. It was featured in Nature, and that got us tremendous attention, and suddenly the psychiatric community started taking an interest in the research we were doing with ayahuasca in my own institution, in Spain, and in many other countries. So, what we found in that study was something really amazing. These participants received a single ayahuasca dose, and some hours after intake, they already had substantial decreases in these scales measuring depressive symptoms. These decreases were around 60% of baseline values. They were significant already on day 1. This is quite unlike the effects induced by traditional antidepressants. And they didn't receive any additional dose or any additional psychotherapy. And these values in these depression symptoms remained below baseline around 3 weeks. That was the last time that we did a follow-up. At 14 days, they increased slightly, but even at that time point, they were around 50% less than the initial values. So what's intriguing with ayahuasca, and perhaps with psychedelics in general, is that they exert very rapid effects, very rapid, in this case, antidepressant effects, and that these effects are sustained for many days after intake. So the research questions we asked ourselves were, why are effects so fast? So it probably has to do with the changes that ayahuasca induces on brain dynamics. And why are they so long-lasting? So we tried to answer these questions by doing an acute administration study to assess changes in brain dynamics, but also studying the post-acute stage of ayahuasca, once the acute psychedelic effects are disappeared around 24 hours later. So probably we think that this long duration of ayahuasca effects may be due to the psychological impact that ayahuasca has on the individuals. There might be also, this had not been explored, neurometabolic changes in the brain. Also, they might be changes in how the different brain regions are related to each other. This has been demonstrated under the acute effects, but not when these acute effects, these acute visionary effects have disappeared. And what we studied as well in this other in vitro study was whether some substances present in ayahuasca could play a role, could have an effect on brain plasticity. So we did studies in lab with cells obtained from animals. So my presentation, I will show you results we have obtained following acute administration in information transfer. This is a metric that assesses the transfer of of information, in this case, electrical oscillations between recording sites. This has already been published. And then in the post-acute effect study, I will show you some modifications in psychological facets, these psychological after effects, or what some call the afterglow. Also results from an MRI spectroscopy study, and also MRI resting state functional connectivity. And in the in vitro study assessing neuroplasticity, I will show you what I think are some amazing effects on neural stem cells, which I hope you find nice. So just to remind you very briefly about the chemical constituents of ayahuasca, you have DMT. This is well known. This is a 5-HT2A agonist. It also interacts with many other receptors. It has recently been found also to interact with the sigma-1 receptor. And it's, as you know, totally inactive when taken by mouth. And when this DMT is combined in the ayahuasca brew with these other substances, with the beta-carbolines, with harmine, harmaline, and tetrahydroharmine, you have a psychoactive effect because these substances are monoamine oxidase inhibitors. THH is also a serotonin reuptake inhibitor. And the new data I'll show at the end of my presentation is showing that they also have these beta-carbolines effects at the neural level. So they interact with neural cells in a different way besides of these monoamine oxidase inhibiting properties or these serotonin reuptake inhibiting properties. So the distribution of 5-HT2A receptor has already been shown by other speakers. There are high levels in the visual cortex. This may explain the visual effects that you obtain with psychedelics in general, but they are also in these two key regions that are part of the default mode network. The default mode network, almost everyone has heard about this. We have this posterior node in the medial parietal cortex encompassing the posterior cingulate cortex, but then you have a more rostral node in this region. So these two areas are rich in 2A receptors, and studies in animals have shown that when you administer a psychedelic, in this case DOI, the effect that you get in pyramidal neurons is an excitatory effect. You get excitatory postsynaptic potentials. And this effect is thought to be mediated by glutamate release. If you administer certain drugs that interact with glutamatergic neurotransmission, you can totally block this excitatory postsynaptic potential. So glutamate may be playing a role in the effects of classic psychedelics. This has been shown in brain slices obtained from animals, but it has never been demonstrated in humans. And it's quite relevant because glutamate is involved in neural plasticity, in long-term potentiation, in the rewiring of the brain, in memory process, in very important cognitive processes that go on in the CNS. And it was glutamate, as you'll see later, what we assessed using this spectroscopy technique. So we'll go first with these effects on information transfer. What goes on here? Why may the effects of ayahuasca be so fast when you administer this to healthy participants or to patients, to depressed patients? And what we saw in this study that we published last year is that there's a dramatic change in the way that the different areas of the brain, or in this case, the different leads that we had placed on the scalp, interacted with each other. So here, subjects were with eyes closed. We were measuring brain oscillations at a series of different sites. And what we saw is that the communication between these sites increased very potently at the peak of the ayahuasca effects, but in a very special way. You have regions in the brain that are high up the hierarchy, basically frontal regions. These regions are usually inhibiting activity in posterior regions that are associated with sensory processing. And under ayahuasca, what you get is the reversal of this transfer of information. It's the lower regions, these sensory selective regions, that start influencing the regions that are high up the hierarchy. So this could also be a way of explaining why people are starting to have all these sensory modifications in the absence of external stimuli. You don't have any external input, you are with eyes closed, you see that there's increase in connectivity, but this increase goes to posterior brain regions, to anterior brain regions. And this is totally the reverse of what you see on the placebo. So this is dramatic enough to think that very potent effects are going on the CNS to induce rapid changes in how the brain is working, and to induce, in the case of this depression study, maybe beneficial effects. And now I'm going to show the new data we got from this post-acute study. So what we did here was assess people before ayahuasca intake and 24 hours later when the acute effects were no longer there. So the first thing we measured was psychological after effects. And what we decided to assess here was any potential increases of what is known as mindfulness capacities. So I have colleagues in my hospital who do mindfulness therapy with their patients. They are psychiatrists, they are psychologists, and they came to me and they mentioned that they saw an analogy in what they had read about ayahuasca and the effects that ayahuasca induced, and what meditation practice, mindfulness meditation practice, was able to elicit. So what we did was administer a series of questionnaires. The first of all is called the 5-facet mindfulness questionnaires, and it assesses different aspects of this mindfulness capacity. And what is being mindful? I can read this definition for you. It says it's a particular quality of awareness that is present-centered, non-reactive, characterized by openness, acceptance, and by a non-judgmental stance towards experience. Also closely related to this idea, it's the decentering or diffusion idea, that is the ability to take a detached view of one's own thoughts and emotions, considering them as impermanent events of the mind. So my colleagues in the psychiatry department had seen that these abilities or these capacities were reduced in many forms of psychopathology. So regardless of what mental illness their patients were suffering from, they all showed a decrease in these capacities. So they wanted to see if ayahuasca would be able to rapidly increase the different aspects that these questionnaires measured. So we administered before and after an ayahuasca session, 24 hours later, these two questionnaires, the FFMQ and the EQ, that's measuring, the last one is measuring decentering. And what we saw were significant increases in these subscales, in the non-react subscale, in the non-judge subscale, in the experiences questionnaire that's measuring decentering. And they had also developed a metric combining those items in these two questionnaires that are usually the most sensitive to meditation practice. So what we found here was that these increases that we were seeing in these three subscales and also in this mind sense composite index was that the post scores were in the range that you obtain from people who have a long practice of meditation. So this left them quite astonished. They start believing that there might be a therapeutic potential in ayahuasca, and this is what has moved us forward in assessing again these facets, trying to link them with changes in brain function. So the second thing we did, this is another study. Again, we administered the same questionnaires, but we did MR spectroscopy. This technique allows you to quantify certain compounds in the brain. So you have to define a region of interest. In our case, it was the medial parietal cortex. We chose this reason for a series of this area for a series of reasons I'll mention immediately after. And what we wanted to measure here was glutamate. As I told you before, it had been shown to be released in these studies in brain slices obtained from animals. And when glutamate is released, it interacts with certain receptors, with AMPA receptors, and it can induce a series of changes in brain plasticity. And in a previous study, we had seen that long-term ayahuasca users showed changes in brain structure precisely in this medial parietal cortex in an area that included the posterior cingulate cortex, and that what they were showing were decreases in cortical thickness. There was a thinning in this region. This region, as I mentioned earlier, is a key node of the default mode network. In the posterior node of the default mode network is associated with self-referential thought, with internal mentation. And in the specific case of depression, it has been observed that depressed patients have a difficulty inhibiting the activity of the default mode network, and that this difficulty is associated with ruminations. So these long-term ayahuasca users were showing changes at this level. These changes correlated very nicely with their lifetime exposure to ayahuasca, and also they correlated with differences in a personality facet called self-transcendence, which is the inclination to have less materialistic and more spiritual views. So for all these reasons, we focused, or we placed our region of interest in the spectroscopy study in this medial parietal cortex. So the idea was that if glutamate is released during the acute effects, what we would observe in 24 hours later in the post-acute stage, in this afterglow stage, would be a decrease of glutamate levels below baseline values. And this is precisely what we observed. We saw a clear decrease in the post-acute stage. So there was a decrease in excitatory neurotransmission in this region. We had administered the same questionnaires I had mentioned before to this new sample of participants, and we saw if there were any correlations between these neurometabolic changes and the changes in the mindfulness questionnaires. So we administered this questionnaire again. There were increases in several subscales, as we had seen before. So here in blue, you can see the pre-administration values. In violet, you see the post-administration values. This is 24 hours later. And in red, you see values 2 months, after ayahuasca intake. So there was a pre-post increase here in these scores. And in the case of this specific subscale, these scores were still elevated, significantly elevated 2 months later. So when we looked for correlations between the decreases in glutamate levels and changes here, in these scores, we see that, again, there appears to be a relationship. So if you decrease excitatory neurotransmission in the posterior parietal, in the medial parietal cortex, this correlates with changes in mindfulness questionnaires. And this correlation is maintained with the difference values 2 months later. So if you calculate the different values between baseline and what you get at 2 months, you still see a correlation with the decreases in glutamate concentrations there. The third thing we did in this study was assess resting state functional connectivity. Maybe you are more familiar with this technique. And we placed a seed in this medial parietal cortex, in this posterior part of the brain. And before ayahuasca, you see clearly that there's a positive correlation. Here, the red-yellow color indicates a positive correlation with these two main nodes of the default mode network. And you can see that these regions are anti-correlated with this area here. This area here is the dorsal anterior cingulate. It's the key hub of the task-positive network. So this is before ayahuasca intake. 24 hours later, we see that the correlation between the posterior and the anterior parts of the default mode network have decreased. The intensity of this connection is decreased. But what is more dramatic here is that the anti-correlation with the dorsal anterior cingulate, with the task-positive network, has totally disappeared. It's not visible anymore. So this is when you analyze each stage separately, when you do the statistical comparison with connectivity from this seed versus the anterior cingulate cortex, what you see is an increase on connectivity. So you are having an increased crosstalk between the default mode network, the task-positive network, in this post-acute stage after taking ayahuasca. And again, we looked, we checked also different possible changes in connectivity. In this case, we placed the seat here in the dorsal anterior cingulate. And what we saw was that in addition to that increased correlation we saw with the medial parietal cortex, there was an increase with the medial temporal lobe in an area that was basically comprised of voxels pertaining to the amygdala and the hippocampus. So here are the voxels that are showing this increased connectivity in the post-ayahuasca assessment. And these areas are well-known regions that are involved in emotional processing, in memory. So you have this in the subacute stage. And when you check for correlations between changes in connectivity and changes in mindfulness facets, again, these interesting results are obtained. Here are increases in the non-judge subscale, and these correlate very nicely with the increased connectivity between the dorsal anterior cingulate and the medial temporal lobe. And the same happens for the non-react subscale of the FFMQ. So this would be 24 hours after drug administration, but you can try to correlate these changes in connectivity with the increases in the non-judge questionnaire, non-judge subscale, that subscale that remained active or elevated 2 months later. And again, there's a clear correlation between these measures. So again, we have a psychological impact that ayahuasca induces. This impact, some facets of this impact are still visible 2 months later. They correlate with the changes we see in terms of neurometabolic activation of certain brain regions and in terms of increased connectivity between regions that are involved in emotion, cognitive control, and memory. And finally, the last data I'm going to show you here is this in vitro studies. So all this that I've shown you is in humans. We are limited by the techniques we can use in humans, and we thought that perhaps something else might be going on at the neural level, at the cellular level, when you administer ayahuasca to humans, and this is what we study. So we did a neurogenesis study. So this what measures is the potential generation or birth of new neurons in the brain. There are two regions in the brain where new neurons are being born even in adulthood. One of these regions is located in the hippocampus, this structure that plays a crucial role in memory and that gets degraded or atrophied in the early stages, for instance, of Alzheimer's disease. We did this in mice. We obtained neural stem cells from the hippocampus. We cultured and we induced these stem cells to proliferate. What's called a neurosphere is formed. And once you get your neurosphere, you add the compounds that you want to test. In this case, it was saline. We tested the beta-carbolines, harmine, tetrahydroharmine. And what you measure after you added your compounds after a certain time is whether these compounds have any effect on the ability, or if they have the ability to promote the early stages of neurogenesis to generate young neurons. This can be visualized using a green staining, an antibody that under the microscope shows a green image. And also, you can measure whether your compounds are promoting neuronal maturation. And this is measured by using another substance that, in these images I'm going to show you, appears in red. So this would be what's called the distal region of the neurosphere. This bunch of neural stem cells cluster. They have a center, and they will have a distant part. And an integral part of neurogenesis is the ability of the newly formed neurons to migrate and to integrate themselves in a circuit. So I'm going to show an image of this distal part in which the newly formed neurons, if any are formed, will have migrated, so that you have additional support for a neurogenic effect here. So the blue spots here are nuclei. So cell nuclei. So you see many blue spots. These correspond to the neural stem cells. And the green staining would show young neurons. The red staining would show mature neurons. So here, almost no neurons are seen. This was what we observed after saline. But when we administered harmine, this is the image that appeared, right? So you can see that it has very potent neurogenic effects, harmine. So these substances that we thought that were only there to allow DMT to access systemic circulation, they may be doing more interesting things than previously recognized. So this left the people in this lab really amazed. I was also amazed when I saw these images. And they also did with this assay with tetrahydroharmine, and the same occurred. So we observed these clear effects for harmine, for tetrahydroharmine, and harmaline did not have any neurogenic effects in this model. So now we are replicating this. This is only qualitative data. We are trying to do quantitative analysis of these findings. And we are also trying to replicate this in vivo with a whole animal. We are administering the substances to the mice, and we are going to see if this also happens in the live animal. So in a time where there is nothing new in sight in the neuropsychopharmacological horizon, most of the drugs we are using are 40, 50, 60 years old now. I think this has a huge potential. This is something we should look into in more detail. And just to give you some potential applications, of course, here I'm just speculating. This is ideas, potential disorders for which we need new drugs. So if we are lucky enough and any of these substances can be used, it will be most welcome. And these include psychiatric disorders like anxiety, depression, addiction, in which structural changes in the brain have been demonstrated, and people have been able to stop taking substances like cocaine or heroin after attending ayahuasca rituals. So maybe part of this possibility that ayahuasca is giving them to change their lives is being mediated by this potential rewiring of certain brain areas. Also, psychological trauma is a field we are interested in. Also, cognitive deficits associated with normal aging, why not? Memory loss that's associated with age, and neurodegenerative disorders for which there are no treatments right now, like Alzheimer's disease, frontotemporal dementia, Parkinson's disease, or Huntington's disease, just to give you some examples. So I'd like to thank again all the people that have collaborated in these studies. Again, Amanda Fielding for her generous financial and intellectual support, and thanks to all of you for your attention.
Thank you very much for your nice presentation, and we are open for one question just. Sorry that we pressed you with the most interesting part of your results.
No problem.
Okay, so please.
I cannot hear you.
It's not on there, or what? Yeah.
Good morning. Thank you congratulations for your presentation and for exciting work.
Go to the mic, please.
Congratulations for the exciting work you have just presented to us. I have a question regarding beta-carbolines and their role in ayahuasca function. Usually, or at least at the first, ayahuasca was thought as some sort of oral DMT. And now we are seeing a lot of evidence showing that harmine has a role, and the other beta-carbolines, either in neurogenesis. There is another paper by Vanya Dakic with these results. And also the work by Eduardo Schenberg, who showed a biphasic effect of ayahuasca depending on DMT and other beta-carbolines. So I'd like to hear from you to elaborate a little bit more about the aspects from DMT and the beta-carbolines, and how could that affect some sort of standardization of ayahuasca for therapeutic uses. Thank you.
In the beginning, I also thought of ayahuasca as oral DMT, just DMT plus something that allowed it not to be degraded in the gut and liver. But gradually, I've changed my vision on this. And although I haven't conducted any direct studies with administering pure DMT, I've talked to many people who have taken DMT and have taken ayahuasca, and they say that DMT is all flashy lights, but very little emotional effects. So my guess now would be that this is not only due to the fact that when you take ayahuasca, DMT is in the system, in your CNS for a longer time, but that the beta-carbolines are also playing a role there. Of course, you have an increase in monoamines because you have this monoamine oxidase inhibiting activity. Also, the blocking of the serotonin reuptake may play a role there. And who knows if what the effects you are seeing in the midterm or in the long term in the regular ayahuasca users is due to this complex interaction of all these compounds. So in this therapeutic studies we are planning to conduct in Barcelona now with the psychiatry department, what we are going to do is administer the combination of these 4 compounds. We are probably not going to use ayahuasca, not even in the freeze-dried form that we've been using in the past, but we are going to administer known amounts of DMT, harmine, harmaline, tetrahydroharmine to try to replicate as closely as possible what you usually ingest with natural ayahuasca.
Yeah, thank you again, Jordi.
Okay.